We are going to move into our 20-minute conversation with doctors Poon, Kaur, and Sosa, just to continue this conversation about small molecule drugs. So my speakers, if you would join me up on the stage. So I'd like to set the stage for this conversation because we've heard three talks that are all pretty detailed and are covering different elements of KIF1A biology and drug development and screening. But this is also an opportunity, if you as a parent or family member have general thoughts or questions about small molecule drug screening, this is also a time to have an open conversation. And so we're hoping that this will be a time for you to ask questions, us to speculate, us to encounter questions that we might not know the answers to, and to come back to them later and continue the conversation. So I'd like to start just by having you talk back to back. I've noticed that in your talks, you were referencing one another and taking lessons from one another. And so I think you're all covering very different elements of KIF1 biology, but there's also relative overlap in the types of ways that you're doing it, the models that you're making. And so I wonder, as you're thinking about your own work and listening to these talks of different elements of drug development and screening, how do you think about that in the context of your own work in ways that you could, strengthen your own systems in conjunction with the other people up here?
I think I can make a start. First of all, I believe in collaborations a lot. So just learning from Dr. Poon and Dr. Sosa, I'm already thinking in my mind, like how if we find a FDA-approved drug which is working on KIF1A, how we can work together to find it more. So again, I think the beauty of this thing is we are working on different types of models. Like we're working in a different strategy, and also looking at different libraries. For example, Dr. Poon is looking at the LOPAC library. We are working on the Compound Australia library, so which is really great. We are not duplicating the efforts, although we are working ahead in finding those small molecules, but it's definitely not duplicative. And I think we can find out the ways that yes, we can work collaboratively if we found some molecules.
Yeah. Certainly, I would love to work with those two libraries and the FDA library to use our molecules. Or structures as templates, and going from there. If we find some that anyone that it combines specifically to these proteins.
Yeah, so I think Dr. Sosa's work is very exciting because that, with the structure in hand, you can really, not fly blind almost. You have a template to then build on structures that might fit and assist or rescue. And so that I think that's very exciting. And I think our efforts to do some of the patient heterogeneity drug assay development, I think Wendy's talk really confirmed for us that we're going down the right path. So that was exciting to hear from our end.
Oh, sorry, I thought I saw yours in your hand. So, Jesse.
Piggyback. Oh, thank you. Yeah. Has KIF1A looked at the Medicanrin AI technology to possibly scope out small molecules that might be cross-referencing each other in the literature that affect kinesin?
So you're asking if we have used AI programs to search the literature and find potential hits? So that is not something that KIF1A.org has specifically done. A lot of our searching is still very manual and seeing what comes up in the literature on a day-to-day basis. Is that something that any of you think about when you are going through looking for potential drugs? What are the ways in which you look for a potential hit as you're exploring the space?
So if on our end, it's these assays are just phenotypes, and so they're phenotypes based on the biology, but we don't necessarily use a biological readout. And so we're looking for compounds to kind of reverse phenotypes, and then and that gets us through identification of a hit, that then we can subsequently test to see if how it's affecting KIF1A biology.
I mean, yeah. So we haven't really started to do in silicon, but we collaborated with through KIF1A with Atomwise. We provided one of the structure that we have, although it was a preliminary one, to do a in silico screen. There are many ways to do in silico screen, some more in depth than other different libraries, but that's certainly something that we want to do, is to use this library and use these structures to see if we can identify a few candidates that then we could try to do tests with them.
I can add a little bit. So in terms of the drugs that we are focusing on, so we think about, first of all, who are the key players involved in the system that we may be able to target. So for example, in our case is the motor protein, and the second is the microtubule. So again, we haven't looked through any AI-based technologies, but that's a very, it's a great point. But what we do is like conventional basis. We go through the literature. We try to find out with some keywords. Has anybody published drug screening, motor proteins? It could be kinesin. It could be other kinesins. Like Dr. Sosa was showing, a big family of kinesin superfamily, which has got the members. So yeah, is there a molecule which anybody has explored has an effect of other kinesins or myosins, or in fact, any of the motor proteins? And is there a small molecule that can stabilize the microtubules, or that can act through making those tracks a bit better and bit receptive, so that even KIF1A is struggling to bind, but they have got some modifications now that it's able to land onto the microtubules a bit better. Yeah, and I think that's a kind of a conventional, but in terms of the library, so we try to pick the library which has got, again, the compounds, the FDA approved drugs, which are being used in other brain related disorders, such that they've got this tendency to cross the blood-brain barrier, and they will be more clinically relevant if we have to use it for KAND later on.
Hi, that was a beautiful set of complementary talks, really helped each other. So I had a question about some of the stuff that was from New Site, which I thought was very exciting. So one of the things that I was struck by is that a lot of the changes you showed were not necessarily related directly, or what you had measured was not in axon transport itself, but various morphological changes in the neurite length or branching and things like that. And so my question was, is this possibly related to the problems in delivery of things like BDNF? And can some of those things be affected directly by providing these growth factors, so as to not only just address the transport issue, but some of the downstream effects of the deficits in the transport of growth factors?
So the reason why we looked at the growth factors is we know that KIF1A is involved in the trafficking of like the BDNF receptor. And so that's what led us down the road of looking at whether or not that was affected or not. While I didn't show it, we have some preliminary data showing that some of our other phenotypes are rescued when we provide BDNF in the culture. And so that is something that we are looking at.
Thanks. It's good.
Michelle. Oh, we got one.
Hi, my name's Jenna Sidhu. I'm from the Michael Silverman Lab at SFU in Vancouver. And I'm relatively new to the KIF1A community and the research, but I was wondering, what are because I work with iPSC-derived neurons, so what are the differences that you see with the different patient variants that you work with in terms of the cell's development and morphology? And then I was wondering if that corresponds to clinical data.
So the question was, how do these differences between different KIF1A mutations show up in the iPSC differentiation from the patient-derived cells?
So the method that NewSite uses to make the neurons is a transcription factor-based approach. So we do bypass a certain aspect of neurodevelopment, of commitment to neuroectoderm commitment. And so we were more interested in once you establish the neurons, how they develop further. And so that was one aspect of the biology that we were interested in. We haven't gone back to dual SMAD, small molecular diff, to see how KIF1A mutations affect neuroectoderm commitment. But that is something that we are interested in.
So from our side, we are still growing our neurons and organoids. So I probably won't be able to speak about that at the moment. But yeah, something to look for in the coming time. But surely we suspect that the variations in the KIF1A is going to impact on the morphology, like how Dr. Poon's work has already shown in terms of its neurites outgrowth. And of course, when it comes to the functional role, especially the epilepsies and the seizures. So yeah, something we would be looking in our system as well.
Hi, I just had a question for Dr. Wayne Poon about the data he showed using the MEA assay, where you first saw like there was developmental delay in the P305L and the 253K, but then I think it was 100 days later you saw that there was an outburst of neuronal activity, if I'm getting that right. So my question was, what do you think could be happening? Why would those phenotypes, those seizure phenotypes show up if what we know about these two specific mutations is that they have a decrease in velocity or, like the E253K, I believe that also affects ATP binding. So there could be decrease in the velocity and run length and so forth. So why do we see like an outburst of neuronal activity if these mutations are actually said to do decreased function, if that makes sense?
I didn't go into this, but there was some interesting work by a colleague of mine, Ann Taylor, at University of North Carolina, where she induced exotomy or axonal damage. And then she identified a retrograde signal back to these excitatory neurons, which actually severed the synapses between excitatory and inhibitory neurons. And that caused then the excitatory neurons to be more hyperexcitable. And so that is something that there are some tools that are being developed to kind of develop assays to look at that. And that's something that we are exploring. And so that is one potential mechanism. I think the second thing that we're also, because we don't want to just put all our eggs in one basket, so to speak, is that we know that in neurodevelopment, it takes about 100 days before astrocytes become fully developed. And so we think that, we have these immature astrocytes, and then after 100 days, they are now fully mature. And we think that KIF1A biology is important for astrocytes, and that's why we've kind of tailored some of our research focus towards astrocytes as well, given the role of KIF1A in astrocytes. And so, I guess a long-winded answer is that we don't know yet, but we have some hypotheses that we're exploring.
That was a beautiful set of talks. I really enjoyed you. I was wondering for Wayne and Sim, who are doing these assembloids or organoids, how consistent of phenotypes you're getting with those? Just in the literature, one of the big struggles that seems to come out of this field is that, even with under control conditions, you get high heterogeneity. And so when you're doing, rather detailed analyses like activity and connectivity of a eye to a brain? I mean, like, what kind of are the readouts that you're getting with your systems like really reproducible? And then, is that tracking back?
So there's, what's nice is that Simran and I have complementary approaches. And so there are pros and cons to the two approaches we have. So our organoids are not organoids that are generated neurodevelopmentally. We just combine cell types at certain ratios of mature brain cell types. And so they kind of maintain that consistency from organoid to organoid. And so this was a strategy that we got from another colleague of mine, Eric Julian at UCSF, in which he was using this for some ALS models. And so he had a lot of RNA-seq data and transcriptome data showing a consistency across organoids. Simran's path is using neurodevelopment to guide organoids. And in her systems, she's going to get the cortical layering that is lacking in just putting cells together. So she might see phenotypes related to how they might project a certain way during development that I'm going to miss in ours. And so we have complementary approaches to make sure that we capture everything that we can.
So our organoids, so for the organoids work, again, like we want to do things in parallel. So again, coming back to the collaborations, we are working with Dr. Anay Gonzalez Cordero from Children's Medical Research Institute, CMRI in Sydney. She's one of the experts who makes the organoids for us, and including KAND, she builds the organoids for a range of different neurodevelopmental disorders, including Rett syndrome and other disorders as well. So from their experience, like you said, there is a lot of variability in terms of getting the structure and then the function, the readouts from the organoids. So to compensate that, we just try to increase the number of the organoids per batch, and we try to do it a couple different number of times. So for example, if we are starting a differentiation today, a week later we'll do another diff, and a week later we'll do another diff. So we try to increase the sample number and the times we are going to repeat the experiment to get more confidence into it. But surely this is something which is very well reported in the organoid world, that there are variabilities within the system. When it comes to the KAND, we don't know yet. So the organoids, they are currently growing. Hopefully by the end of the year, we will get more answers for the readouts. But I think based on their experience, they are getting pretty consistent readout if they take care of the number of the organoids that they are bringing into the analysis.
Hi, actually along the glia line, because we also use both neurogenin 2 differentiated neurons, as well as we just are moving into the small molecule differentiation. And one of our reasons for doing that was because we were worried about skipping over like normal developmental events, like you had mentioned. And with the virally transduced cells, you do not get glia cells as you would with the small molecule differentiation. So then we usually introduce glia into our culture. So then I was wondering about what you mentioned with glia with the mutation. Have you done cultures with glia without the mutation and with the mutation, and have you seen an effect on the development of those neurons?
So we hope to we're going to be poised to do that experiment probably at the beginning of the year. Right now, the wild type astrocytes are ahead of the KAND mutant astrocytes. So we'll be there soon.
This is a beautiful question. And when we talk about understanding the biology more, like how Dr. Poon was saying, we need all those, the orchestra of the different cells which are supporting the neurons. So we definitely want to look at the neurons along with the glia cells, including the astrocytes and microglia, and especially because of their role in the neuroinflammation part as well. So when it comes to the organoids, the system that we are using, it's called the spontaneous differentiation method. So the astrocytes, they get automatically, so when the organoids are developing, the astrocytes spontaneously start differentiating within the organoids. But in the organoid world, the microglia's penetration or the generation of microglias within the system that we are using is pretty hard. So even if we supplement the system with the microglias, they try to remain on the boundaries, but the penetration, the deep penetration within the three-dimensional ball-like cells, it's a quite tricky. But we have found alternative ways to deal with that, something which is very similar to what Dr. Poon is currently suggesting. So there is a special chambers in which we can grow the neurons along with the astrocytes and the microglias in like a jelly kind of a thing, so that we can kind of help them support and mimic like a three-dimensional structure. So there are ways to kind of do about that. But we are also very looking forward to Dr. Poon's work, in which he's having like the astrocytes derived from the KAND patients, and then he's going to supplement the neurons. So it's something which is very exciting. And we were having a chat this morning about how about the microglias. So this is something, yes, but very interesting point. Thank you so much.
So it was very interesting to learn how you put together the aggregates by taking defined proportions of different cells. So I was curious in that context. I think it was, you said that after 100 days, there was a change in the behavior of the cells. So is it possible to monitor that heterogeneity to know whether that transition is related to a change in proportions, whether there's a particular degeneration of a particular cell type, or what's the critical transition correlated with?
So that's a great question. And we do have an experimental design for that, where we are over 100 days with that transition. We will be able to collect the cells and do some experiments to look at that. I think, without going into the nitty gritty, one of the things that is a challenge with MEA is that, at least with the system that we're using, you can't image the cells simultaneously because of they don't have a clear bottom, at least for the Maxwell platform. So it's been a challenge to get corroborating data on what's happening on the electrodes. And so, we've been working with those companies to try to find different solutions to allow us to do that experiment.
Possible to monitor the electrical activity with like calcium sensors or something that would perhaps allow you to do imaging of multiple things simultaneously?
That's a very good question. So we are exploring calcium sensors for that as well as potassium channels. So yes.
Such a wonderful questions. Thank you. And coming back to your question, Ajay, about correlating the electrical activity with more structural information, like what's going on. Is there a change in the cell proportions? So this is something that Dr. Poon recommended us to do, considering that organoids, they will have those structural layering with time. So I think the plan would be we will be growing a couple of number of organoids within one well. So hopefully they are acting similar because they are getting similar environment. Some of them, they will be measured on the MIA, but the others, what we will do is we will take them, we will section them, and then we will put some antibodies for a key proteins, just to kind of, first of all, get more information about, is there any structural changes? And of course, something about the change in the excitatory and the inhibitory neurons ratio, which thanks to Dr. Poon, this is what we want to kind of highlight as well, that we are always, even though we don't speak so much in person, but then we are always on email exchanges and we are exchanging ideas. So I really appreciate that collaboration, and we look forward to work. And of course, if anybody has got more suggestions about the systems or what we can do with what we are already doing, so we will be very happy to do that.
And I think that's a wonderful note to wrap up this discussion on. Obviously, our wonderful speakers will still be around for these conversations, and we hope you do take advantage of that.